welcome back, and let's start the second part, which is learning about reducing and non-reducing sugars. So we talked about how acetyl and hemiacetyl structure are formed after attacking by ROH group, which is alcohol group. So there are two different tests which is required to understand whether your sugar is reducing or non-reducing. So one is reducing sugar and other is non-reducing sugar. Okay. So reducing and non-reducing terms are um, referred in describing the properties of sugar. So you have two different things, two different tests to see whether your sugar is reducing or non-reducing, which is reducing copper or silver. Okay. So the reagent we use for copper is Felling's reagent. Okay, and this test is called Felling test. Okay, and in this Felling test you have a copper with second oxidation state to copper in first oxidation state. Okay, and can you tell me where the oxidation state of copper is decreased or increased? The copper in the first stage is +2 oxidation state and the second is +1, so it's decreased or increased? Decreased. Okay, so anything that can decrease the oxidation state is a reducing reagent. Okay, can you be a reducing molecule or can be a reducing compound? So here any sugar which can reduce copper from its second state to its first state would be a reducing sugar. Okay, and if they have a positive for Felling's test, they are reducing sugar. If they get a negative of Felling test, it's a non-reducing sugar. Okay, this positive and negatives are color changes in the reaction. Okay, and the other thing you can do is another test which is tolerance reagent, and this is called tolerance test. When you have a silver reduced from copper +1 to copper 0. Okay, and if you have any sugar molecule that can reduce these two, Felling's reagent and tolerance reagent, you call them as reducing sugar. Okay, if they cannot, they are non-reducing sugar. Okay, and how they can or they cannot is hidden in their structure. Okay, so we'll understand how. Let's see some important derivatives of hexose. Okay, up to now we learned about the simple structures of glucose or simple structures of fructose. Okay, now these are modified forms of glucose and fructose. Okay, so the first one, I think you still remember that beta out, and it's beta D glucose, right? The second one is N with a NH2. NH2 is for amine, and that's why we call beta D glucosamine. Okay, can you relate this? This has a word amine. This has a word amine. See, this amine is for this one. Okay, and if you draw the number of carbon 1, 2, 3, 4, 5, and 6, you can say that glucose are 2 amine because second position is replaced by amine group. Okay, can you see this is an acetylamine? This group is called acetyl group. Okay, this is called acetyl group, and this has a amine, so that's why we call is an acetyl glucosamine. Okay, this is still beta D glucose, and this N is attached to acetyl group, and that's why called an acetyl beta D glucosamine. Okay, did you get how they have done the nomenclature? Similarly, beta D galactosamine, amine group. So this amine comes from this because they have amine group in the structure. Okay, the most famous is N-acetyl glucosamine. Okay, be called as NAG, and other is N acetyl mm, N acetyl muramic acid. Okay, NAG and NAM. They are two different famous N-acetyl glucosamines, which is derivatives of hexose. Okay, so N-acetyl glucosamine and N-acetyl muramic acid. Okay, but they are derivatives of same glucose structure. Okay, so it's easy to remember. Just you have to know what is what, and this is if they have additional R group, and R group is this. This could be called as muramic acid. If you have R group and N-acetyl muramic acid. Okay, these are derivatives of beta D 6 phosphate. Okay, you have 6 prime 6 carbon replaced by a phosphate group. Okay, I could beautifully teach you glycolysis, Krebs cycles, and oxidative phosphorylation using these structures. They are beautifully arranged one by one. They phosphorylate, dephosphorylate, and forms other structures, their isomers, and so on. Okay, if you have learned glycolysis earlier, it starts with glucose 6 phosphate, fructose 6 phosphate, dihydroxyacetone phosphate, glyceraldehyde, and so on, and the reaction goes to form phosphoglyceric acid, which is PGA. Okay, and then so on. So you just have to remember the structures over there, but not here. But this is uronate, which is this group, and it's open D-gluconate. This lactone form, this is important. So most of the antibiotics have this ring, which is called lactone ring. Okay, but anyway, you just have to remember those structures with a different replacement of either hydroxyl group or R group or something else. Okay, not very important, but you can practice it at home. Glycosidic bond, one of the most famous bond in sugar, like peptide bond in protein. Okay, glycosidic bond, this is between two glucose molecules, that's why they are called as glycosidic bond. Okay, so whenever you have glycosidic bond, you have a acetal structure. What is acetal structure? We already have gone through how to make acetal bond. Okay, and we'll see different disaccharides. Suppose maltose has 1,4 bond, somewhere we have 1,6 bond, and we'll see how we can make 1,4 or 1,6 bond in disaccharide. Okay, the word here comes is disaccharide. Di means two, saccharides mean sugar, saccharine, sugar, saccharide, sugar. If you have two sugar molecules, it's a disaccharide. If you have many, then polysaccharide. Okay, so this is how they are known. If you have just one, then monosaccharide, disaccharide, polysaccharide. Okay, so this is how you name them. Let's move to the next part to understand what is the 1,2 and 1,4 structure. Now, can you remember this hemiacetal structure? We already have drawn this somewhere. Whenever you have hemiacetal, means you already have one group left, you can reduce them. Do you remember that? If you do ROH, it will again replace this into OR. You remember this? Okay, so if you have a OH group, it means it's a hemiacetal. Okay, and if it's a acetal structure, means you have no R group left. Okay, so this acetal structure, and this would act as alcohol, and if you do the condensation reaction, it will remove the water. This is OH from here, H from here, see, and you will get a oxygen here connected with both of them. It's similar to how you remove water molecules when you add two amino acids and make a peptide bond. You still remember CONH2 and COOH, OH and H, and you remove to make a peptide bond. This is similarly, you remove hydrogen from one glucose and hydroxyl group from other glucose, and there will be a removal of water molecule to form a acetal bond. Okay, and this bond itself will be called as glycosidic bond. Okay, this bond is called as glycosidic bond. Okay, but you have to name them specifically depending upon the number of carbon. This is first carbon, this is second carbon, this is third carbon, fourth, fifth, and sixth carbon. Okay, if you see this one, next to this is your first carbon. Okay, always remember, next to oxygen is your first carbon, always and always and always. Okay, next to your oxygen is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and your CH2OH will be always sixth carbon, last carbon. Okay, so can you see them? This is your first carbon and this is your fourth carbon. So I can say this is C1 C4 glycosidic bond. Okay, do you see how I name them? C1 C4 glycosidic bond. In short, we write 1,4 glycosidic bond. Okay, cool. If you see this structure, this is again a hemiacetal structure, which means they have a OH group that can be replaced, and that's why the sugar is reducing sugar, because they have a hemiacetal after polymerization. Okay, if you see a OH at the first carbon, and first carbon is anomeric carbon, you remember that first carbon is anomeric carbon, and if you have a hemiacetal at anomeric carbon, your sugar is still reducing. Okay, and if you have acetal structure at anomeric carbon, your sugar is non-reducing, which we will learn after this. Okay, am I clear up to here? Yes, no? Do I have to repeat this? Let's understand one more time how we make glycosidic bond. Okay, again is non-reducing sugar, I said, which have a glycosidic bond, but they have acetal structures in glycosidic bond, not glycosidic but anomeric carbon. They have acetal structure, they are non-reducing sugar. Okay, non-reducing ends, they have non-reducing ends, that's why they are non-reducing sugar. We'll learn how we can see. Trehalose is an example of this, and we'll see the structures of that. Okay, I'll do one by one to compare and make you understand what do you mean by reducing and non-reducing sugars. Okay, the first thing is, let's understand what is this molecule and what is this molecule made up of. It's all galactopyranose and glucopyranose. Okay, now we are going in more complex structural forms. Do you remember glucopyranose? It is made up of glucose, and this galactopyranose is made up of galactose. Okay, I'll check your memory what you have learned before the break. Do you remember this galactose? This galactose is epimer of glucose, but at what carbon? 4 carbon, 4G, remember? So galactose is 4 carbon epimer. Can you name the carbon number? 1, 2, 3, 4. Can you see a difference from glucose to galactose? Let's name them 1, 2, 3, 4. And if you remove this bond, you will have oxygen and hydrogen here. So can you see at fourth position, your hydroxyl group is different, and that's why they are galactose, right? Are you following me? Yes or no? Mannose is 2 carbon epimer of glucose, and galactose is 4 carbon epimer of glucose. That's why you see a difference of H and OH at 4th carbon if you draw the structure of galactose, right? So now when the bond is already formed, and this bond is known as, anyone? Glycosidic bond. Okay, but can you name this bond properly? This is carbon 1 and this is carbon 4. That's why they are called beta D galactopyranosyl 1,4 beta D glucopyranose bond. Okay, this 1,4 is basically carbon 1 and carbon 4. Okay, in short, we write gal beta 1,4 glucose. This gal is for galactose and GLC for glucose, and this beta is represents these both are beta forms. Why? Look at the anomeric position. There is a OH which is out. Out means beta. In means alpha. Okay. You see this beta because its OH is out. Okay. Both OH is out. That's why they both are beta D glucose, beta D galactose, and beta D glucose. Okay. And this is how you name them. Can I move on to the second structure to see what we have? Let's understand. This is 5-member ring. This cannot be glucose. This is fructose. And this is 6-member ring. It could be glucose. Let's understand 1, 2, 3, 4, 5, 6. Okay. Between one carbon and second carbon. Okay. So this is C1, C2 glycosidic bond. Right. C1 and C2. Okay. I said whenever you write oxygen here, this is second, third, fourth, 5. 6 will always be CH2OH. So the orientation of fructose is the other way. Okay. Do you see that? The orientation of fructose is the other way as of glucose. Right. And this is the second carbon. This is first, second, third, fourth, 5, 6. This is 6 carbon. Can you see a hemiacetal group anywhere? Can you see a hemiacetal group anywhere? Hemiacetal means OH at an anomeric carbon. This is anomeric carbon. Does it have a OH? No. This is anomeric carbon. Does it has a OH? No. That's why they are non-reducing sugar. Okay. Can I compare this with the first one? Yes or no? Can we say lactose is a non-reducing sugar? Brainstorm. This is anomeric carbon. Carbon 1 is always anomeric carbon. This is anomeric carbon. Does it has a OH? No. Does this have a OH? Yes. Means this is a reducing sugar. Okay. Can you see this? This is anomeric carbon. Does it have a OH? No. Does it has a OH? No. It means they are non-reducing sugar. Can you call sucrose as non-reducing sugar? Yes, sir, you can, because there is no OH to be reduced. Okay. And sucrose is a disaccharide made from alpha D glucose and beta D fructose. Okay. And the number of the way they are attached is glucose alpha 1 beta 2, alpha 1 beta 2. Okay. The name, number of carbon, and the way hydroxyl groups are present. This is alpha D glucose. Why? This structure is alpha. This is alpha D glucose. Why? Because OH is inside. This is beta D glucose, beta D fructose. Why? Exactly, because the hydroxyl are on the other side. You see it in because this is opposite structure. Okay. But this is how you see beta and alpha. Okay. Another, I wouldn't say whether it's reducing or non-reducing. You have to say this is reducing or non-reducing. Also, the number of carbon is different. It's 1 to 1. So one alpha glycosidic bond. Can you see that? One is glucose, other is also glucose. So two glucose molecules attached with 1 and 1 carbon are trehalose sugar, which is a disaccharide. Okay, can you see that? Is it a reducing sugar or non-reducing sugar? Reducing. Why? Because they have a OH group. Yes, you are completely wrong. So this is a, so I always told you, look at the anomeric carbon. Okay, What is anomeric carbon here is next to this. Does it has a OH group? No, sir. Does this one has a OH group? No, sir. This one is not anomeric carbon. Okay? If you are confusing, let's see the other structure. Look at this. This is anomeric carbon, yes. But this one is not anomeric carbon because this is not next to oxygen, right? Am I clear? So this is a non-reducing sugar. This is not a reducing sugar. Okay? Cool. So I think up to this point you understand what is a reducing sugar and what is a non-reducing sugar. Okay? If you do not want to remember this, just remember the name or whether they are reducing or non-reducing. Trehalose is a non-reducing sugar. Sucrose is a non-reducing sugar. Lactose reducing sugar. So you just have to remember the names and then you're good. Okay. I think we are finished up to disaccharides. We finished monosaccharides, we finished disaccharides, and now we'll finish polysaccharides. Okay, cool. So poly means many. Okay, many could be same or different. Okay, what do you mean by different? So imagine if we have 10 Apple phones, all are same, apple, 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 apple. So this is homopolysaccharide. Polysaccharide made up of the same thing. Okay, other we have heteropolysaccharide. Hetero means different. Okay, if you have Apple, Samsung, Motorola, I don't know what other companies, but yeah, you have different monosaccharide units attached together to form a polysaccharide. Okay, and they are called as heteropolysaccharide. Okay, homopolysaccharides and heteropolysaccharide. Okay, same units or different units. Okay, cool. So see, homopolysaccharides all are yellow. Who's a fan of Coldplay? Okay, so all are the same yellow, and then it's branched. It could be unbranched or it could be branched. So homopolysaccharides. Homo means same, poly means many, saccharides means sugar. Okay, homo means same, poly means many, saccharides means sugar. So homopolysaccharides. This is heteropolysaccharides. Hetero means different, poly means many, saccharides means sugar. Heteropolysaccharides. Okay, see, these are different colors, 1 2 3, no 1 2 1 2 1 2 1 2. Okay, but there are only two monomers, but there could be multiple, means 1 2 3 1 2 3 1 2 2 1 3. Okay, so this could be two or many, but they are heteropolysaccharides. Okay, this is a 6-membered ring, right? It could be glucose, it could be any other molecule which is a monosaccharide. Am I clear up to here? Yes sir, no sir? See, we have glycogen. Glycogen is most famous one. Why? We all have glycogen in our body. If you have more muscles, you have more glycogen. Not necessarily correct. You could have more muscles, but if you have more glycogen, you have more strength, because you can have more energy hydrolyzed from that glycogen. Okay, and the glycogen you store them in liver and muscles, right? So glycogen is also a polysaccharide, but they are made up of glucose, because you need glucose for energy, and all of them are made up of glucose. They are monosaccharides of glycogen. Okay, can you tell me confidently that you know what is alpha 1,4 and what is alpha 1,6? It's the position of glycosidic bond. You see this 1,4 and 1,6, 1,4 1,6 1,2, it could be anything. So if you see glycogen, it has 1 4 and 1 6. 1 6 are branched and 1 4 are just linear monomers. Okay, I'll show you what is 1 6 and 1 4 later on. Okay, but this is it for now. So they are storage polysaccharides in animals, and we are also kind of animal, social animal. We do store glycogen in our body. Let's understand starch. What is the main source of starch you already eat? Do you know a starch you eat? What's your name? Texas? Proud Texan. What's the starch you eat? You love to eat? Yes, it's the same thing you're thinking of. Yes, but what is it? What starch do you love to eat? Yeah, you're right. I'm telling you. I can read your brain. But what is it? What's the starch you love to eat? Tell me, you're correct. Yeah. But what is it? Tell me the name of the starch you love to eat. Come on. We are in Texas. Any starch you love to eat? Exactly. Potato chips. And anything else? Any starch do you love to eat? Corn. Corn is a good source of starch. You might have seen labels high corn fructose syrup or something in packages, which is not healthy, but I see that. High fructose corn syrup, right? Corn is a good source of starch. And any other famous thing you eat almost every day, which is mostly starch? That's why you say don't eat starch, don't eat carbohydrates, because it will make you fat, right? Reduce carbs, more protein, right? Rice, we eat a lot of rice and it has a lot of starch. So rice, potato, anything they have starch, but they are plant stored glucose, and glycogen is animal stored glucose. Okay, so see, nature has made two type of glucose in animal and plant so that they can store them differently, and you eat them and you get the same glucose in your body. You can utilize them for your energy, right? Isn't it beautiful? They have alpha 1 4 linkage having amylose and amylopectin, which is branched. Okay, in glycogen you have seen 1 4 and 1 6 linkage, and they also are branched, but they are present in oh, stop mirroring. UGLC, can you tell me the code? 0899. Okay, so plants stored glucose is starch, animal stored glucose is glycogen, right? Okay, the next is glycosidic linkage in glycogen and starch. Let's see how it's different. Can you see? Let's understand amylose, and this is amylopectin. Okay, amylopectin, amylose or amylopectin. Okay, so I talked about alpha 1 2 linkage, alpha 1 4 linkage, alpha 1 6 linkage. Okay, these are three types of linkage we have seen. 1 1 also at some point for tree halos or something. Okay, but can you see they are all glucose? Yes. Okay, can you see this is a glucose molecule? And when you see a starch, they have two different forms. One is reducing, one is water soluble, and other is water insoluble. Okay, so have you ever washed rice before cooking? So when you wash, or potato before cooking, if you slice potato, dip them in water and start cooking, that will have more sugar, less sugar, if you wash them before cooking. So whenever you wash potato or rice, you see a white color, uh, not like, it's not transparent, it's more translucent. You see a watery fluid with white color. those are all water soluble starch you removed before cooking. So you have less starch if you cook your potato or rice before cooking. Okay, and all those non water soluble is already there. So it's a good way to remove your calories if you wash your potato or rice before cooking. Okay, so they are in two different forms, which is amylose and amylopectin. Okay. Oof. I need to be continuous. Okay, once again. Okay, cool. So this is how a mixture of amylose and amylopectin look. If you see amylose, they are never branched. They are unbranched structures of starch. And if you see amylopectin, which is this one, they have a branch. This one, see, this is branched. They are interconnected to each other. Okay, and branch points are alpha 1 6, and unbranched are attached with alpha 1 to 4. Okay, so they have alpha 1 4 linkage and they have alpha 1 6 linkage. Okay, alpha 1 4 linkage and alpha 1 6 linkage. Anyway, did you understand this one or no? If they have a branched point, this is basically 1 6 linkage. Okay, if you do not understand, see here, you have a you have a monomer as polysaccharide, and you have a branch from this to this, and this is 1 6. Okay, so branched polysaccharides in starch or glycogen has 1 6 linkage. Okay, this is how they represent. This is branch point. There will be no no sugar from here. Okay, it will start from here. So that's how they are represented in this way. Okay, once you have a branch, your monosaccharides are over after that. Okay, so now see metabolism of glycogen and starch. Okay, starch are present in the form of granules in plant cells. Glycogen and starch both are in granules. They contain enzymes that synthesize and degrade polymers. They have many non-reducing ends. If you see the structure, you could see reducing and non-reducing ends. Enzymatic process simultaneously occurs in many non-reducing ends. These are mostly information. Cellulose. Cellulose is another polysaccharide, very famous. Okay, and it's very important for your diet. It doesn't give you a calorie. That's why people say eat more salad. Salad has cellulose. Mostly plants have cellulose in their stir form. We cannot digest cellulose because we are not ruminants. If we could digest cellulose, it will be really a big problem for us, right? Eat anything that has a plant base, you will get cellulose, will be in your body, and you will get more and more calories, right? So that's why if you eat more cellulose, there is no enzyme to digest those cellulose to make glucose out of that. Okay, that's why whenever you eat salad, they do not give you any calorie. Okay, they are roughage in the form of your food, and it's very good to have them because you will not get calorie even after eating them. Okay, so they are another polysaccharides, and they have alpha 1 4 linkage. You just have to remember them. They make hydrogen bonds, and that's why they are stable. Structure is now tough and water insoluble. If you dip salad into water and wash them, you will not see any watery fluid, white fluid, because it's not water soluble, it's water insoluble. And most abundant polysaccharide in nature. Okay, most abundant polysaccharide in nature is cellulose. Okay, most abundant, this could be a question. Most abundant polysaccharide or most abundant carbohydrate is this one, cellulose. Okay, which is present in plant tissues. Okay, cotton is purely most this is a purest form of cellulose. Cotton, your clothes, cotton clothes are cellulose. So if you eat them, you will not get any calories. Or even the woods, they are cellulose. If you eat woods, you will not get any calories. Okay, So these are two different examples.